I request our respected principal sir, Dr. Shivdas Sirsat sir, to give us some guidance.
Good morning, everybody. Marathwada Shikshan Prasarak Mandal's Yeshwantrao Chauhan College, Ambajogai, Department of Microbiology, organized one-day national level seminar on recent trend in microbiology. I am grateful to all Honorable Executive Council and CDC members. The office bearers of all organizations have given their best wishes for this seminar. Today, our guest of honor and keynote addresser of this seminar, Honorable Dr. A.M. Deshmukh sir, President of Microbiologist Society, India, and Professor and Head, uh, Department of Microbiology, Dr. Babasaheb Ambedkar Marathwada University, Subcenter Dharashiv, resource persons, and our senior colleagues, Professor Dr. Pushpalata Jadhav ma'am, Professor and Head, Department of Microbiology, Devgiri College, Chhatrapati Sambhajinagar, my all teaching and non-teaching staff members, convener of this seminar, Miss Nikita Jadhav, and organizing committee members, technical supporter. I have an immense pleasure to organize these events for the benefit of new building scientists, research scholars, and faculty students. Dear participants, recent trend in microbiology, trend in microbiology provide a multidisciplinary forum for the discussion of all aspects of microbiology from all biology and immunology of genetics and evaluations and rangers across virology, bacteriology, and mycology. Microbiology is at present one of the fastest moving disciplines in life sciences. Recent trends in microbiology are focused on addressing raising global challenges such as climate change, food insecurity, and emerging health threats. Some major threats in research field of microbiology can be mentioned. Human microbiome research and personalized therapies, genetic engineering, synthetic biology and microorganism, environmental microbiology and climate change, anti antimicrobial resistance and mechanisms, phage therapy revival, artificial intelligence in microbiome research, mobile and real-time microbial monitoring. Lastly, I appeal to the participants to the online throughout these events and take the benefit of the deliberation from experts. I hope that there will be fruitful discussion in this seminar. Once again, thank you all. Jai Hind, Jai Bharat.
Okay, thank you, sir. It's a really an honor for our college to organize such a beautiful program with such an amazing guest, Dr. M. Deshmukh sir and Dr. Pushpalata Jadhav ma'am. Words are too short to describe their achievements. We are feeling so fortunate to have both here today. Then I request to Poonam Lakhe ma'am to introduce our today's guest, Dr. M. Deshmukh sir. Poonam ma'am. Poonam ma'am.
Good morning. May I audible to everyone?
Yes, ma'am.
Hello.
Yes, ma'am.
Okay. Good morning. Good morning to everyone. Dr. E.M. Deshmukh sir was working as a president in Microbiology Society in India and also professor and head of a microbiology department at the Babasaheb Ambedkar Marathwada University, Sub-Center Dharashiv. Sir has 37 years of experience. He has successfully guided 17 research students and one working how they pursuing the PhD work. under the guidance of the sir.
चाबी काढ.
Chairman of a board of studies in the microbiology, member of a board studies, research and recognition committee of environmental committee. Also, sir talk on the radio to awareness was created regarding the biofertilizer, biopesticide, pure water and vermicomposting environmental pollution. Sir worked as a nanotechnology, biotechnology solution to e-waste and biogas and the dye degradation. Most important thing about the sir, that is he made many informative video in the COVID-19 period. Thank you so much, ma'am.
Okay, thank you, Poonam ma'am. To know more about recent trends in microbiology, we would like to call respected Dr. M. Deshmukh sir to deliver speech on the topic recent trend in microbiology. Deshmukh sir.
Hello.
Yes, sir.
Dr. A.M. Deshmukh here. So, whether I should start?
Yes, sir.
Okay. Thank you. Thank you. Thank you. So, at the outset, I'm very happy today to interact with the students of Ambe Zogai from Yashwantrao Chauhan College. I'm really thankful to Principal, Honorable Principal, Senate Member, Dr. Shivdas Shirsat sir, for encouraging the faculty to arrange such academic interactions which are useful to the students, which are useful to the society, and useful to the all. Again, at the beginning, I should take the name of Dr. Nikita Zadov. She was regularly in touch with me. She was asking my convenient time, and today she has organized this event. The title of the event is a recent trends in the microbiology. Okay. Now, whether the slide is visible to you?
No, sir.
Okay, 1 minute. I will share it. Because, yes. But again, I would like to tell all, now visible.
Yes, sir.
That although I am sharing the slides are not important for me. I want to talk, I want to interact with you all, and I want to tell that microbiology in the present scenario is one of the important science at a global level. One example I would like to give you, that on 24th August 2024, exactly 6 to 8 months back, Honorable Prime Minister of India, Narendra Modi ji, he has passed one bill in the cabinet. The bill was regarding Bio E3. Bio E3 meaning is that biotechnology for 3E. Which are the 3E? One E was employment, another E was for economy, and third E was for environment. And in his lecture, he told that his lecture is still available on the Google. He told that in coming next 75 years of this century, biotechnology will dominate at a global level. And I am also supporting his statement. I am telling that now for the next generation, two technologies are very, very important. At the top, there will be artificial intelligence, and next to artificial intelligence will be biotechnology. And in the biotechnology, 90% is a microbiology. Meaning is that microbiology will dominate, and there are many, many recent trends. Although we go back 300 years when Antony van Leeuwenhoek first time shown the presence of microorganism. But if we go in Ayurveda, the presence of microorganisms have been shown by many rishis, many doctors, Ayurvedic doctors, 2000 years back, 2500 years back. Indirectly, they have shown the presence of microorganisms. Now, it is our time to review that what are the new trends, recent trends in microbiology. We know the bacteria, fungi, algae, actinomycetes, all other names which have been taught in the classes. But although they are taught in the classes, nowadays there are many, many recent things in the microbiology, and our students should know. One example I would like to give you, that in India, the cervical cancer for the woman is a very, very important. Very important means that many women die or suffer because of the cervical cancer. Then WHO report says that 7 lakh ladies per year are suffering by ovarian cancer in India. Now understand, out of the 7 lakh ladies, death is a very few. But all these 7 lakh ladies has to be admitted in the hospital. The operation should be carried out to remove the ovary, then radiotherapy, then chemotherapy. They require minimum 6 months stay in the hospital. More than 10 lakh rupees, sometimes maybe 50 lakh rupees. And then they will live normal life. So to prevent it, Microbiology Society has written a letter 1 year back to the Public Health Minister of India. Again, I am coming to the recent trends. Why? Before 10 years, there was no any treatment to the cervical cancer. There was no vaccine available for the cervical cancer. But later on, the cervical cancer vaccine was developed by the Western countries. Although it was developed, the cost was very high, non-affordable to the common man. The per dose price was 3,000 rupees, and the minimum 2 doses were required to give to the girls. So 6,000 rupees for the vaccine. So common man was reluctant to take that vaccine. We have written a letter to the Honorable Health Minister of India. And what is the effect of it? 6 months back, Honorable Nirmala Sitharaman declared that now this vaccine will be given free of cost in public health center of the government, PSC of the government, free of cost. You see, what is the price if you go privately to the market? Price of that vaccine is only 300 rupees per dose. These are the recent trends. In the microbiology, many vaccines are playing very important role. And common man should know what are the recent trends in vaccine. Again, I am telling, if you remember the corona was caused by virus. But how the corona was controlled? Corona was controlled because of vaccine developed by virus. Meaning is that the problem is a virus, solution is a virus. That is the power of microbes. So, in the recent trends, you must tell to the young generation, you must tell to the adult person, you must tell to the ladies, many vaccines are there which are available in the market. Again, one more I will tell you. The vaccine against pneumonia is available. Every person above 60 years age should take the pneumonia vaccine. But there is no awareness. And many old persons die because of pneumonia in elder age. One example I will give you. Honorable past President of India, Pranab Mukherjee, died because of the pneumonia. There are many, even, you see, Sindhu Tai, ma'am, died because of the pneumonia. Pneumonia is a very dangerous and vaccine is available. There is a vaccine for TB. There is a vaccine for hepatitis. There is a vaccine for mumps. There is a vaccine for measles. There is a vaccine for rubella. There is a vaccine for corona. Like that, more than 20 to 30 vaccines are available. And we must take this vaccine. Whenever we are talking about the recent trends in microbiology, vaccines role is indispensable. And very fortunate part, all these vaccines are produced in India. Means India is very strong in microbiology and supplying these vaccines to the world. Again, one more thing I would like to tell you in recent trends. Just Honorable Principal have told about he has used one word, antibiotic resistance. It is a recent trend. What is antibiotic resistance? You see, before 10 years, before 20 years, there was no antibiotic resistance. And before 1954, there was no antibiotic available in the market. Question doesn't arise about the antibiotic resistance. First antibiotic, you know, all of you, history, that first antibiotic was developed by Alexander Fleming, penicillin antibiotic. Although it was discovered, although research was carried out for 20 years, understand, in 1928, he has shown the presence of penicillin antibiotic. Up to 1942. No one was knowing the importance of this antibiotic. It was the first Hitler. Someone has told to Hitler that our soldiers are suffering by wounds. What he has told? Our soldiers are suffering by wounds and sepsis. And some Alexander fellow is there. He has proposed one antibiotic. Why we should not request to Alexander? Then the Hitler first requested Alexander and told him, what is the solution you have? Alexander told that, give me conical flask, glasswares. I am having fungus with me. I will propagate it. He has propagated penicillium fungus. And the cell growth he was rubbing on the wound and soldiers were recovered. That was the first application. Now, what is the situation? Again, I am coming today, recent trends. Today, antibiotic resistance is a major problem. Not only in India, but out of the India. But I want to tell you that out of India, let us take example of UK. the application of antibiotic is carried out rarely. Antibiotic is not available in the medical shop. Any person cannot purchase antibiotic without prescription of doctor in UK. What is the situation in India? In India, the situation is that in any medical shop, you can go and purchase an antibiotic without any prescription. This is a very wrong. I have given a letter to the government, central government, even to the state government. Then state government has agreed, accepted suggestions given in our letter. But all these suggestions are on the paper. The government is telling that in every shop there should be CCTV, medical shop, and there should be check without prescription. Medicine should not be given, but in practice it is given. It leads to the antibiotic resistance. And I know one MP from Nagpur, Satav, he died because of the infection of antibiotic resistant bacteria. See, meaning is that he was rich with the money. Many doctors were at his disposal. Everything was with him, but doctors could not survive only because of the infection of antibiotic resistant organism. Nowadays, I will again warn you, all students, in the future year, during next coming 10 years, any of your relative admitted in the hospital, there is a danger that he will suffer from antibiotic resistant bacteria. It is a very common in the hospital infection, and there is no treatment. There is no remedy. Only we have to see the death of our close relative because of the antibiotic resistant organism. This resistance is developed, and now scientists says that there will be next epidemic will be of antibiotic resistant bacterial infection. So whether we should wait for it? For that purpose, what we have to do? We have to stop the antibiotic resistance and create an awareness among the society. Now again, because you see, I will be talking about 40 minutes. And within these 40 minutes, it is not possible for me to cover all aspects of recent trends. But I want to focus on some important aspect of recent trends. Now, the maximum research carried out throughout the world and in India is about the gut microflora. And some students might be knowing Microbiologist Society has started one campaign, drink buttermilk and stay healthy. Why? Again, I'm linking it with the recent trends. Now, the research is carried out on gut microflora. And it has been proved that the microorganisms in your intestine and in my intestine are very, very important, playing important role in maintaining your good health. If the microflora in the intestine is disturbed, example, you are suffering from infection, doctor gives you oral antibiotic. Whenever you are taking antibiotic through the oral route, all organisms present in the gut will get killed. To re-establish the microflora, doctor is giving tablet which is available in the medical shop, probiotic tablets. So instead of that, I'm telling drink buttermilk. In the buttermilk, probiotic organisms are present. Which probiotic organisms? Lactobacillus, leuconostoc, streptococcus, bifidobacterium, and many other organisms. All these organisms are essential in the gut. If they are disturbed, why they are disturbed? They are disturbed if you take drinks. They are disturbed whenever you are taking the beverages, carbonated beverages. They are disturbed whenever regularly we are drinking tea. They are disturbed whenever we are eating antibiotic, taking antibiotic, as well as they are disturbed whenever we are taking excess of starch through the pizza, through the noodle, and such starchy substances. So, if you want to maintain, drink buttermilk. Your microflora in the intestine will be maintained. What role it plays? It is called as a recent trend sampling, fecal oral therapy. Nowadays, it is a very common fecal oral therapy. You go in the hospital, big hospitals. If anyone is having weakness, his intestine is a poor intestine, he has many problems, doctor gives him microorganism through the oral. Which microorganisms? The microorganisms isolated from the fecal matter of the healthy person. Are you getting it? Microorganisms isolated from the fecal matter of the healthy person. These microorganisms are establishing in the intestine, and these microorganisms improve the health of weak person. Are you getting it? This is the recent trends in microbiology. Gut flora, common intestinal flora plays important role. Not only in the physical health, the CFTRI, Mysore, Central Food Research Institute in Mysore, they have shown that these microorganisms also produce neurotransmitter, meaning is that which goes and improve your brain health. So, 1 minute. They improve your mental health. They make you happy because of the microorganisms which are present in the intestine. They make you fresh, avoid depression. That is the advantages, and it has been proved. These are the recent trends in microbiology. Again, one more recent trend I will tell. Bioplastic. All students should know it. What is a bioplastic? Because you know that plastic has been discovered, and later on it was observed that plastic has many uses because of the elasticity. The bags are prepared from plastic. Tubes are prepared from plastic. Many parts in medical equipment are prepared of plastic. Then sheets are prepared from plastic. Many things are prepared from the plastic, and this plastic is a non-degradable nuisance to the environment. So what scientists are searching? Scientists are searching biodegradable plastic, and whether they are successful? Yes, let me tell to the student. Whenever there is a search, there is an invention. The bioplastic, one lady from BSc third year microbiology from the Ahmedabad, she discovered bioplastic. How? From the saga. Saga in Marathi we call it as a sabudana. It is elastic in nature. She is having her own industry of preparing bioplastic from the saga. Likewise, some are preparing bioplastic from potato. Some are preparing bioplastic from the bacteria. Bacteria are producing polylactic acid and poly-beta hydroxy butyric acid. It is a bioplastic. There are many organisms which are producing bioplastic. This bioplastic is in market. You go in the market and ask, we want a disposable bag, they will give you. But there is no awareness. Today's lecture, why I'm giving marks to your college, today's lecture contribute in creating awareness among the students. There are plastic bags which are biodegradable, and these should be replaced. These should replace the normal traditional plastic which is used. So, bioplastic is a recent trend. Vaccine is a recent trend. Then antibiotic resistance is a recent problem. These are the recent trends. Now, one more thing I want to tell you, single cell protein, SCP. Now, what is SCP? These are the blue-green algae. See, it is a BGA, and this BGA, spirulina, it is observed that it is green in color. If it is cultivated, dried in powder, and if we eat it through orally, it is the best nutritional food on the earth. It is having higher protein content than egg white. It has higher protein content than soybean. It has all amino acids. It has many vitamins. So, spirulina is a very, very powerful. People are not knowing. Even doctors are not knowing. I'm going to tell you, whenever weakness is there, doctors are advising you eat the eggs. It's correct. But if the person is a vegetarian, even then they are forcing, although you are a vegetarian, you must take eggs to survive. Why you are asking to take eggs? You tell to that patient, take spirulina. But doctors are not knowing spirulina. Spirulina is a wonderful food. It is called as a single cell protein. It is available on the market, Amazon. You go on Google, search it. Capsules are so costly. One capsule costs 15 rupees, 20 rupees for one capsule. But it is highly useful, spirulina. Now government also understand. There are many industries. I'm telling to the student, why you are searching for a job? You start any industry, spirulina industry. There is a good market. One of my students is in Pune. He told, sir, I got an order from the central government, defense ministry, that we want sweets containing mixed with the spirulina. We got a 50 lakh order. You see, all these are recent trends. What are the recent trends? The sweets are available now in the market. All these sweets, all bakery products are there. All chocolates are there. You students may not be knowing there are spirulina chocolates available in the market. These are the recent trends. Our microbiology students should be proud of it, that the biotechnology and microbiology has captured a market. That's why Honorable Prime Minister is telling that now in the India, bio business will dominate. Everywhere you will see that who is earning money, bio business person will be earning money. Microbiology person will earn money because of these recent trends in microbiology. Not only in that, now some industrialist has developed a culture. Which culture? Culture for biodegradation. These microbial packages are available. You go on in Google, search it. The pesticide degrading bacteria. Then lignocellulose degrading bacteria. Cellulose degrading bacteria. The many other compounds which are non-degradable, which are called as a genobiotic. All these can be degraded by bacteria and the cultures are available. They are making money. by selling their microbial culture for degradation. Again, one more thing I would like to tell you, genetically modified food. You see, you are a students of microbiology. You should know what is the present status of the genetically modified food. I would like to tell you, genetically modified foods are not allowed by the parliament in India. Indian parliament has denied. They will not be allowed. What is the situation in Canada? What is the situation in America? What is the situation in UK? They are allowed. They are eating the food, but Indians are not allowed. Otherwise, what is the genetically modified food? Genetically modified meaning is that one bacterial gene is transferred in the plant. Which gene? Cry gene, crystal gene, from Bacillus megaterium. From the Bacillus megaterium, cry gene is taken from bacteria and added in the plant. What is the advantage of it? The advantage is that whenever we grow that plant in the field, any worm cannot attack on it. And the production will be more. The plants will be protected from bollworm, from different types of insects. If they eat the leaf, small piece of leaf, they will die. That is called as the magic recently carried out by the microbiologist. That's why it is called as a recent trends. So, genetically modified foods are allowed even in Bangladesh. See, Bangladesh government has allowed BT brinjal, but India has allowed only one crop, that is the BT cotton. Other than BT cotton, no other crop is allowed. It is available. All tests has been carried out. In the Jalna, there is a one big biotechnologist industry. He is having very big biotechnology industry. It is a Barwali. They have carried out all tests. They are ready to produce, but government is not giving permission. So, why I'm telling you, I have written many times to the central government. Please allow genetically modified food so that the food problem will not be there in India and our microbiology students will get jobs in many industries. But government is not allowing the genetically modified food in the market. Then come to the biofertilizers and biopesticides. I think it is approximately 40 minutes are there. I will talk for 5 minutes more, correct? And then there will be question and answer. So, the microorganisms in biofertilizer and biopesticide. Now, whole world is telling that don't use chemical pesticides. They are poisons. You know the very famous film star Amir Khan. He conducted one episode. And in the episode, he told that the chemical pesticides kill human being. Don't use chemical pesticides in the field. Ultimately, they will enter in your body and my body through the animal milk, and we suffer from the means health hazard, different problems, maybe cancer, maybe any other problem because of the chemical pesticides. So, what is the alternative? Farmer will ask, what is the alternative? Alternative is a biopesticide. They are available. Example, trichoderma. In Nashik, one fellow is selling trichoderma, earning lakhs of rupees. Trichogramma is there. Tricho cards are available in Bangalore. And these tricho cards are sold in the market. They are biopesticide. There are many biopesticides are available in the market. Now it is a time to tell to the farmer, don't use chemical pesticides, use biopesticides. They are not harmful to you. They are not harmful to the animal. They are not harmful to the birds. Because of chemical pesticides, many birds are dying. Many animals are dying. So there is a need for the creating awareness regarding these recent trends in microbiology. In addition to that, I want to tell you, now the recent trends tell that to clean the environment, the microorganisms are important. You see, the microorganisms are important to clean the environment. How? The electronic waste. Before 20 years, there was a very small amount of electronic waste in India. At present, electronic waste is created in tons and tons. Mobile waste is there, laptop is there, discarded laptop, computer, unused computers, and many other electronic equipments are thrown out on the garbage, and they are the best. If we want to recover different metals from this garbage waste, and if we want to prevent a nuisance entry of these metals into environment, biotechnological solution is there. We can recover all these metals with the help of microorganisms. So you see, microorganisms are very powerful. They are used in every field. Which field? You take in agriculture, in health also. In health, microorganisms are playing very important. At an industrial level, you take any fermented food. You see, any fermented food, the microorganisms are playing important role. Pickle, idli, dokla, and many others. In that, the new microorganisms are now added in this food material to increase the taste, to improve the flavor. There are many recent trends also in food microbiology. In addition to that, in medical microbiology, vaccines and antibiotic resistance are playing very important role. I have told you regarding HPV vaccine. Every girl below 14 years should take HPV vaccine. It is freely available in the public health center, but we don't know. We are microbiologists. Even then, we are not knowing. Common man, layman, how he will know what are the recent things going in the microbiology. In addition to that, there is a bio cement available. There is a bio steel available. Understand? Bio decoloration process is available. There is a maximum research going on in bioinformatics and many other things. So the cement prepared with the help of microorganism, is it in market? Yes. If you ask me, sir, we want more light, more focus. You go on the YouTube, write down bio cement, and one of my student, Dr. Trapti Jamalpure, she has worked on it, and she has to produce bio cement with the help of microorganism. So you see, to all students, again, I will stop here. And I will request all students, do maximum awareness among the society regarding the recent trends in microbiology. That is indirectly, it is a social service equal to the soldier which is doing social service at the boundary of India and Pakistan, at the boundary of India and China. So all microbiology should be proud of it, that microbiology has many things to do in the society. Thank you again, Nikita ma'am. Thank you, Dr. Pushpalata ma'am. And thanks, Honorable Principal Sir, for giving me an opportunity to interact with the students. Thank you. Thank you very much.
Thank you so much, sir, for your informative and valuable guidance. It will be definitely helpful for us. Okay, then I request Poonam ma'am to introduce our next speaker, Dr. Pushpalata Jadhav ma'am. Poonam ma'am.
Hello.
Yes, ma'am.
May I audible to you, ma'am?
Yes, yes.
Okay. Good morning, everyone. Now I will happy to introduce Dr. Pushpalata Jadhav, ma'am. She is a professor and head of a microbiology department in MSP Mandal, Devgiri College, Sambhajinagar. Ma'am has a 20 research papers are published in national and international journals. Actually, ma'am has a lot of achievements, but uh, we are tell you in short because of a time limit. Ma'am are working in examination duties, paper evaluation, also paper setting as a chairman for the third year. Also elected as a BAS member in a microbiology subject, expert and examiner in the poster presentation on the occasion of a biocentenary year of a scientist of the Louis Pasteur. So we will very eager to listen such a great personality. Thank you, ma'am.
Thank you. Thank you, Poonam. Thank you very much. Am I audible?
Yes, ma'am.
Yes.
Yes, ma'am.
Yeah. Respected Principal Dr. Shivdas Shirsat sir, Senate Member Dr. Babasaheb Ambedkar Marathwada University, Chhatrapati Sambhajinagar, my mentor and guide, respected Dr. A.M. Deshmukh sir, President of Microbiology Society India, retired Professor and Head, Department of Microbiology, Dr. Babasaheb Ambedkar Marathwada University, Subcampus Dharashiv. All delegates and dear student participants, good morning. Now it is good afternoon to all of you. Today, MSP Mandal's Yashwantrao Chauhan College, Ambazogai, District Bid, Department of Microbiology has been organized virtual one-day national seminar on recent trends in microbiology. I congratulate Dr. Shirsat sir and Miss Nikita Jadhav madam. Department of Microbiology has been arranged a nice session today. Today, I will deliver my lecture, a very important topic, biofertilizers, production and their applications. I choose this topic because last decades, the use of agrochemicals on the soils, there is a decreases in the fertility of the soil. Whatever the native microorganisms, beneficial microorganisms which are important for the crop plants, they are decreasing, and it is very toxic for the environmental, increases the environmental pollution. So I choose this topic, very important for the student also and very interesting, biofertilizers production and their applications. Nikita, can you share the PPT?
Yes, ma'am. Wait a minute. Yes, ma'am.
Yeah. Next slide. Now, first we should know what is the biofertilizer. Everybody know that biofertilizer is actually the living solution of the beneficial microorganism, which are very important for the crop plants. It is selected strains, maybe azotobacter, maybe rhizobium, maybe azospirillum, maybe azolla vam, a live culture of the microorganism which is spreaded in the soil, which increases the fertility of the soil and which increases the availability or uptake of the nutrients for the plants.
Sir, next slide.
Yes, continue केलं तरी चालेल. Now, first, biofertilizer and microbial, it is nothing but the microbial inoculants. Actually, it is a live organism. Biofertilizers, why we used for the crop plant as compared to the agrochemicals, which increases, which causes the toxic effect on soil. It decreases the fertility of the soil, and it is very harmful to the microorganisms which are indigenously found in our soil, and which has the ability to increase the nitrogen fixation, which increases the crop plants. So biofertilizers are the low cost renewable sources of the plant nutrients, which improves the soil fertility and crop productivity. So ideally what they do, they maintain the soil health. Very important, maintenance of the soil health means increases the fertility of the soil, minimize the environmental pollution. If you use the biofertilizer directly live solution on the soil, seed solution, whatever it may be, it minimize the environment pollution. Though there is no harm to the pollution, it minimize the environment pollution. It is eco-friendly. So it cut down the use of chemical fertilizer. We cannot use agrochemicals or the chemical fertilizers on the soil. Next slide.
Sir,
So if we go for the commercial history of the biofertilizer, at first it was launched on nitrogen as a laboratory of culture rhizobia in 1895, and in India the first study and commercial production of rhizobium was found by Dr. V.N. Joshi sir in 1956. Some government inventions are there. National Fertilizer Development Center are there. It is situated in the Ghaziabad. Central Sector System is there, NPD, that is the national project on the development and use of biofertilizers. Financial assistance increased from 13 lakh to 20 lakh per unit if you use for the biofertilizers. Government plays a dominant role in marketing by three ways. That is, first is state government via district level officers and village level workers. State marketing federation via cooperative bodies and farmers. State agro industries cooperative via agro service center. Now in India also last few decades there is increasing the biofertilizer production. State-wise, if you see in Tamil Nadu there is a more production of biofertilizer. As compared to Maharashtra produces very less biofertilizers. Karnataka, Tamil Nadu, and Gujarat has the produces the more biofertilizer, and there it maintains the environmental sustainability. Biofertilizer production increases the crop yield and also increase the economic value of the crops. If you go for the classification of biofertilizers, biofertilizers are divided into the 6 group. Nitrogen fixers are there, azola, next is the phosphate solubilizing microbes, vesicular arbuscular mycorrhiza we called as VAM. Plant growth promoting rhizobacteria, nowadays we call it as the PGPR. Sulfur solubilizing microbe, these are the 6 types of biofertilizer. Along with this, nitrogen fixers are again 2 types. In that there is a bacteria are there and the blue-green algae. Azola include the Azola filiscoides and the Azola rubras. In bacteria, we already know that the very important biofertilizers are produced mainly from rhizobium, azotobacter, mycobacterium, isospirillum, bacillus, etc. And in a blue-green algae, that is a blue-green algae we called as BGA. It includes the Anabina, Nostoc, Tolyprothis, Anavinopsis. These are the biofertilizers mainly which are used for the production of more crop. If we use this, there is a increase in the fertility of the soil and gives the more crop yield. The one tabulated form of the number of fertilizers are there, biofertilizers, and the fertilizers, what is the contribution and how they are beneficial for the particular plants. Now the nitrogen, first nitrogen fixer is the rhizobium. We already know it is a symbiotic nitrogen fixer and it has the ability to fixes the 50 to 30 kilogram nitrogen per hectare. It leaves the residual nitrogen, increases the yield by 10 to 30%, and maintains soil fertility. It is very important aspect which increase the fertility of soil. And what are the plants get benefited from the rhizobium? Pulses legume, it includes the cowpea, green, black gram, oil legumes, fodgrums. These are the plants which get benefited by the biofertilizer in which the rhizobium is used. Second and very important biofertilizer example is the azotobacter. This also supplies the 20 to 40 gram of nitrogen per gram of carbon source. It has the ability to promotion of the growth substances like vitamins B group, indole acetic acid and gibberellic acid. Again 10 to 15% it increases the crop yield. It also maintains the fertility of the soil, and it has the ability to biological control of plant disease and suppress the plant pathogens. If some fungus or any other substance get infected the plants, it has the antibacterial or anti bacterial activity against that fungus. So biological control of the plant disease, the azotobacter is very important. Which plants get benefited by the azotobacter? The mustard is there, sunflower is there, banana, sugar grapes, papaya, watermelon, tomato, ladyfinger, coconut, spices, flower, crops, and forest species. Now, the third and very important biofertilizer we can use, that is the azospirillum, which has the ability to fixes the 20 to 40% kilogram of nitrogen. Results increases mineral and water uptake. Vegetative growth crop yield. And what type of plants get benefited by this? Rice is there, sugarcane, finger millet, wheat, sorghum, bajra, etc. And the next and very important is the blue green algae. That is 20 to 30 kg nitrogen per hectare in submerged rice field. Production of growth substance like auxins, indole acetic acid, and gibberellic acid. These are the growth substance which are necessary for the growth of plants. So some bacteria has the ability to produce this, synthesize this. So azotobacter is one of that biofertilizer, and one of the bacteria is the blue green algae, which has the ability to produce the growth substances like auxins, gibberellins, or the indole acetic acid. Which the plants get benefited by this is the rice. And the last and very important again, that is the fifth one is the azolla, which has the ability to fixes the more as compared to the other, that is the 40 to 80 kilogram of nitrogen per hectare fixes by the azolla and use as a green manure because of the large biomass. The size or azolla is large, so the biomass is more, so it can be used as a green manure. And which plant is benefited? That is the rice. So role of biofertilizers, we already know that it makes the availability of the nutrients easily. What happened, the plants are unable to fixes the atmospheric nitrogen by themselves. So microorganism or bacteria play very important role, make that nutrient available for the plants. So very important, first is the makes the availability of nutrients to the plant, very important. Make the root rhizosphere more lively. So rhizospheric region of the plant should be lively. Growth promoting substances are produced. Just we said that azolla and azotobacter has the ability to produce the indole acetic acid is there, gibberellin is there, auxins are there, which has the ability to increase the crop yield. These growth promoting substances are very essential for the crop yield. So these are called as the growth promoting substances which has the ability to produce by these azotobacter and azolla. More root proliferation, better germination, improve quality and quantity of the product, improve fertilizer use efficiency, more biotic and abiotic stress tolerance, improve soil health. So if we use the agrochemicals frequently in that farming, so it decreases the fertility of the soil. and there will be no health, soil health. So there will be no more crop yield. So if you use the biofertilizer, it improves the soil health. It increases the fertility of the soil, and ultimately it gives the more crop yield due to the use of biofertilizers. Residual effect is there, make the system for more sustainable. So we already said that is it is a eco-friendly. If we use the biofertilizers as compared to the agrochemicals or chemical fertilizers, it is more beneficial and it is eco-friendly. So in encapsulation, when we say that the use of biofertilizer it is a live culture of the microorganism in the liquid form. If we spread in the soil, if we soak in the seed, and then it is spread at a particular field of the farmer. For that, if the culture is live, to improve their self life, bioencapsulation process is very important. So it is called as the immobilization of microorganisms, so which increases the field efficacy and it increases the self life of the biofertilizer. So it is one of the step in that. So these are the process how there is encapsulation can be carried out to immobilize the cell. So biofertilizers in the market, they are sold in a particular liquid water, that is which is sterilized water, and in the form of solution. It is given the expiry date, everything is written on that, and it will be sold in the market. So azospirillum is available, phosphate bacterium is available, azotobacter is available, azorhizo is available. So these are the biofertilizers can be sold in the market. These are the packet or the packing form of the biofertilizers. So this is the how there is a production in the large scale biofertilizer. If you have the culture of the rhizobium, if you have the culture of azotobacter, azospirilla, azolla, how will you get the more amount of the biofertilizer in large scale, which is used or to supply to the farmers? Sir, next slide. First, so this is the stages of development of the biofertilizer. First, in which bacteria we are interested. Either it is a azotobacter, either it is a azospirillum, either it is a rhizobium. So isolation of microbes from the roots or the soils. Laboratory screening of microbes for plant growth. So first we already know the micro soil contains the millions of microorganisms. We are interested in the only beneficial microorganism or the those have the ability to increase the fertility of the soil, such as the azotobacter or the azolla or azospirillum. So these bacteria can be screened out in the laboratory. And then after screening, greenhouse screening of microbes to promote the growth in the potted soil. Whatever the microorganism gets screened by using a proper culture, the selective culture for that bacteria, it gets screened out in the laboratory in small scale, and then it used for the potted soil. After potted soil, the field screening of most effective microbes in the crop soil. Once you get the culture of the bacteria, which is the useful as a biofertilizer, that can be used field screening of most effective microbes in the crop soil. And then refinement of the inoculums, environment impact test, and substantiation of microbes, and then there is the production. Very important is that first you have to isolate the pure culture of that bacteria, either the azotobacter, azolla, rhizobium, and then it is in the small scale, it can be grown in the flask level, and then it is subjected for the fermenter in the large scale. And then after the packaging, it can be sold in the market. This is the mass production, that is the bacterial culture subculture into the nutrient broth. That nutrient broth gives the more amount of bacterial cell, and then culture again shake on the incubator shaker at 30 degrees centigrade. 30 degrees centigrade, and the culture incubated until it reaches maximum cell. Near about it give the more population of bacteria such as 10 raised to 10 to 10 raised to 11. And then optimum condition, the population level could be attained with 4 to 5 days for rhizobium. Rhizobium culture should be kept for 4 to 5 days, azotobacter for 5 to 7, azospirillum 5 to 7 days, and for 6 to 7 days azotobacter. The culture obtained in the flask is called as starter culture. Now, if you get such a type of culture in a 4 to 5 days or 5 to 7 days, whatever the culture we get in that flask, we called it as a starter culture. So for large production, inoculum from start culture is transferred to the large flask fermenter and grown until the required level of the cell count is reached. So we need the cell count 10 raised to 10 to 10 raised to 11. More population of the desired organism should get, and then we are subjected for the large fermenter. So media used for the mass culturing is again very important to get the desired organism or the pure culture of the respective bacteria. So if we use the rhizobium as a biofertilizer, for that we can use the yeast extract, mannitol agar, congruent medium. If we use the azospirillum, Dauberniers malic acid broth with sodium chloride. If we use the azotobacter, Waxman number 77 broth, Phosphobacteria picovexki broth, Pseudomonas king's bee broth, and Trichoderma pdi, that is a potato dextrose broth. All these broth, whatever the culture is written here, all are selective for the respective media. Then and then we get the more culture of the bacteria, then it is give the large population of microorganism, and we can produce the large scale biofertilizer of respective bacteria. So if we use the selective media for that respective bacteria, we get the pure culture of that microorganism, and there will be no chances of the contamination. So I have procedure, how the appropriate culture we taken into the broth, and a respective bacterial cell is an aseptic condition, incubated at 30 degrees centigrade for 5 to 7 days in rotary shaker. The continuous aeration is very important. So the rotary shaker is important. And then observe the culture and estimate the population, that is the starter culture. We say that is a 10 raised to 10 to 10 raised to 11 large population of that respective bacteria. If we get, then and then we supply it for the large quantities in the fermenter. Again, this is sterilized and cooled well. That media is fermenter is inoculated with a log phase. A log phase, we know that in that phase the microorganisms are actively metabolized, and they are active in that phase. So such a culture, if we took for the production of the biofertilizer, it will give the more yield of the biofertilizer. Cells are grown in fermenter by providing the aeration and continuous stirring. All the bacteria are aerobic, so we have to provide the continuous supply of the oxygen for the growth and multiplication of the biofertilizer bacteria. For that culture is continuously bacteria get aerated and continuously stirring is very important. So broth is checked for the population of inoculated organism. So all this process is very important. After aeration, continuous stirring for 5 to 7 days, the population of microorganism must be checked for whether it is a pure culture or not. So it must be checked for population of inoculated organism. And cells are harvested with the population. Load is 109 cells per ml. So the more population of microorganism we require, as we are interested to get the more biofertilizer. So carrier material is very important to adhere that microbial cell to that carrier material, which is very inert, and good quality of fertilizer we get from the carrier material. These carrier material, they are inert, they are non-toxic, non-chemical, cheaper in cost, locally available, high organic matter content, no toxic chemical, water holding capacity of more than 50%, and easy to process. So such a carrier material are used for the production of biofertilizers. So these are the carrier materials for the respective bacteria. These carrier materials increases the shelf life of certain bacteria and it retain for long duration of time. If we are interested to prepare the azotobacter, so inoculants packet are very important. Preparation of inoculant packets. So first neutralize and sterilize the carrier material, spread and clean dry sterile metallic or plastic. Bacterial culture drawn from the fermenter is added to the sterilized carrier and mixed well by the manual mechanical mixer. Inoculants are packed in a polythene bags sealed with the electric sealer. So that carrier material are mixes with the culture and then they are sealed. So these are the specification given for the polythene bags. First, it should be written by the name of the manufacturer, name of the product, strain number, the crops of which recommended, method of inoculation, name of the manufacturer, batch number, date of expiry. You see here important is date of expiry. When we say that it is a live culture of the bacterial cell, so expiry date is very important. After that we cannot use, it may lose the efficiency of that bacterial culture, act as a biofertilizer. Then write the price, full address and storage instruction is very important again. So these are the control standard for the biofertilizers. That is the control at various stages: production, marketing, and application. Quality of inoculation, that is a success or failure, acceptance or rejection. On the basis of the pure culture of that bacteria, it can be decided either it is accepted or rejected, and then they are supply for that production, marketing, and application. So he already that is the selection of the bacteria is there. First, there is a regular inspection of the quality control and authority of the under the acts. Identification of strains guaranteed, genus and species, very important. Now, when we are interested to produce a particular bacteria used for the production of the biofertilizer, identification of that strain is guaranteed. What is the genus? What is the species level? Density of the strain should be more, that is a 10 raised to 9 to 10 raised to 11. If such a quantity we get, then and then it is effective, otherwise not. Assessment of main activity as effective indicators of biofertilizers. So you can try on pot basis and then you can use in the larger scale. So these are the some acts which are used for production of the biofertilizer before it's sold in the market. Evaluation of effect or target crops. So whatever the biofertilizer bacteria we took, either the rhizobium, either the azotobacter or azolla, for the specific bacteria the specific plants are given. So it first trial on that plants. If it gives the more yield, then and then it can be used for the production of the large scale and the registration under the regulation. Biofertilizers application methods. So first is the seed treatment, that is pelleting, root dipping treatment, seed treatment, and soil application. If once we get the large amount of solution of the bacteria bioinoculant of that culture of that biofertilizer, how will we apply? What are the treatments? How will we get the more crop yield? For that, first is the seed treatment is there. Second is the root dipping is there. Seed treatment is there. Soil application. These are the number of methods how to apply that biofertilizer to get the more crop yield. So for the seed treatment is very easy. For that, most common method in that is the it is used for all types of inoculant actually. Seed treatment is effective and economic. So the seeds are treated with biofertilizer and kept for shade 30 minutes and then seed become ready for sow. Actually what happened when we say it is a live culture of the bacterial cell. Live culture of bacterial cell itself is a biofertilizer. So that solution, the seeds get soaked, and after soaking for 30 minutes, they are ready for used for the sowing in the field. Set treatment, in the set treatment it is used for the sugarcane. Cut pieces of potato and base of the banana suckers. Prepare the culture of suspension of 1 kg of biofertilizer with 40 to 50 ml of water. That is, ratio should be 1:50. And these sugarcane cut pieces of potato and that they are dip in that, and then they are used for the in the field. One of the method is the seedling dipping. In the seedling dipping method, uprooted, it is used for the water dipped in solution of the biofertilizer and kept in at least 20 minutes and transplant immediately. Ratio is 1:10. That is a one. If you get the 10 ml of solution of the biofertilizer, 100 ml of the water should be there. For root dipping, dissolve 1 packet of the biofertilizer with 20 liters of water, 200 to 300 plants it is necessary, and 1 packet in 2 liters is sufficient to treat the 200 to 300 sets for the cutting method. How to directly apply on the soil? The mixture of biofertilizer plus compost plus soil applied on land before sowing of seed or transplanting to the main field. The mixture of biofertilizer and cattle manure soil sprinkled with water is then broadcasted into the soil at the time of sowing or at the time of irrigation in standing crop. So once the biofertilizer, compost and soil mixture is there, they directly used in the soil, and then they are soil sprinkled with the water is then broadcasted into the soil in the time of sowing or at the time of irrigation in standing crop. So it is very important to of the liquid biofertilizer. So liquid biofertilizer already said that is the seed treatment is there, soil application is there, biofertication is there, and injection into the soil. Seed treatment, you already said that is the seeds are soaked for 30 minutes in the live culture of the biofertilizer. Soil application, directly you spread the culture of the inoculant on the soil. Biofertication, the sugarcane sticks are deep into the biofertilizer and then they sow. And the injection into the soil, the biofertilizers, liquid culture of the biofertilizer, injection of that directly at a particular area should be added into the field. Some disadvantages. This is the last point in the biofertilizer production. Even though we have said there are the too much advantages of biofertilizer, increases the fertility of the soil, it is eco-friendly, there is no harm to the health of the human being. So even though it has the advantages, some disadvantage should be there, but it is not health hazardous. What are the disadvantages? Biofertilizers require the special care for long-term storage because they are alive. So the culture is alive. So the storage and the caring is very important to sustain, to remain for longer duration of time. Must be used before the expiry date. So when it is a live culture of bacterial cell, so expiry date must be there. Before expiry date, we can use. If other microorganisms contaminate the carrier medium of growers or wrong strain, they are not as effective. If the pure culture of the biofertilizer get contaminated, there will be no use to use as a biofertilizer. And biofertilizers lose their effectiveness in the soil is too hot and dry. If the climate is hot and the soil is dry, then effectiveness of the biofertilizer will be less. So these are the very important topics already fall into the recent trends in microbiology. Today, the students, all microbiology students are attending this virtual conference. So I took this topic. It is fall into the recent trends in microbiology. Biofertilizers production and their application. It is a burning topic in today's decade. So most of the students of microbiology can start their startup, can start their business By taking the culture, we can grow in our laboratory, and we can use in the larger scale. We can earn the money from that. So it is very important as a microbiologist, at a microbiology student, to aware the farmer, to aware the society to use the biofertilizer rather than the agrochemical. So this is the topic of the today's recent trends in microbiology. Thank you very much. My sincere thanks to Mr. Principal Sirsat sir and Miss Nikita Zada, who has given me this opportunity as a resource person to deliver a talk on very interesting topic. Thank you, sir.
Thank you so much, ma'am, for valuable information regarding recent trends in microbiology. It will be definitely helpful for us in future aspects and also in your day-to-day life. Okay, thank you, ma'am.
Okay,
Then,
thank you.
then would I like to call Kople ma'am for vote of thanks. Kople ma'am. Kople ma'am.
Yes. Good afternoon, everyone. I am here to offer my thanks on behalf of all the participants. We would especially like to thank Dr. A.M. Deshmukh sir and Dr. Pushpalata Jadhav ma'am, our chief guest, for accepting our invitation and joining us today. Your presence has boosted our energy and your thoughts have inspired us all. Your words have really motivated us. I appreciate our principal Dr. Shivdas Sirsat sir and vice principal Dr. Ramesh Shinde sir for always encouraging and directing us. And thank you Ghodke sir, our science coordinator, or who always support us to work hard and do our best. I also want to thank all of you for joining and helping us to make this day and successful and special for all of us. An event is unsuccessful without a loving and appreciating audience. And also thanks Asif sir and Ali sir. All people who are attending here deserves many thanks, and thank you all of you.
Once again, thank you, Ananal participant, for your support.